Hierarchical cluster analysis is a method for grouping similar objects in a dataset, and it's used by heat maps to show patterns in high-dimensional datasets. The algorithm is very simple, and it's based on the similarity of groups of objects. So each object starts out as a cluster, and we measure the distances between the clusters, the pairwise distances, and then merge the two that are the most similar. So in this figure, samples 1 and 2 get merged for having the same shape and the same color, and they become cluster number 5. Now we're going to measure the distance between cluster 5 with cluster 3, cluster 5, and 4. We already have the distance between 3 and 4, and we're going to merge the two that are the most similar. So we repeat this. We're going to combine 3 with the first two, with this cluster 5 to come up with cluster 6. And then when the final merge happens, and there's still one cluster remains. So the observations that are at the bottom of the tree are the most similar, and those that are at the top are the most different. So this is an example of what we call a greedy algorithm. Greedy algorithms are problem-solving approaches that make the best local choice at each stage of the analysis. They're often not optimal, but they can get really close, then they're usually applied because they're easy to implement. So the question is, how do we define a dissimilarity that occurs between groups of objects for building our tree? So we need a definition. There are 4 different methods I'm going to show that apply, and I'm going to apply those definitions to the same data set, and you can see how the tree shape changes based on your choice of dissimilarity. So the first one is called single linkage, and it's also known as nearest neighbor linkage, and that's because the distance you compute between the groups is the minimum distance from looking at the pairwise distances between points from different groups. Now the tree that comes out is based on often elongated and has irregular clusters. And the second approach is complete linkage. This is known as farthest neighbor. So it takes the points that are the farthest apart and has the distance. The bias of this approach is that it tends to find compact clusters, and the method's susceptible to outliers since it's focusing on combining farthest points. So the third method is average linkage. This one's also called group average method because you're going to take the distance between all pairs of points, one from each cluster, and average over all of them. So each point in one cluster, the distance is then computed with every point in the opposite cluster, and you do that for all the points and get the average. So it tends to find clusters with the same variance. Since it considers all cluster members, it's less influenced by extreme values than, say, the complete linkage or the next method that I'm going to tell you. And the last one, this is called Ward's minimum variance method. This is a statistical criteria where it's going to minimize this error sum of squares term. So this is like a term from an analysis of variance, where if you have cluster centers, say you have k clusters, you're going to compute the distance of sum over all features, your observation's difference from the mean of all the values in the cluster. That difference is squared, summed over features, and then summed over clusters. It tends to merge small clusters, and it's biased towards creating clusters that have a similar size. And because you've got this squared of the distance, it can be very susceptible to outliers as well. Now, these dendrograms, they're sensitive to several choices we make. So one is the dissimilarity metric that I just described, those different choices you have. Another is whether or not you standardize the variables that go into the data or not, that go into the data set. Outliers, I mentioned those as being influential. And then the variable, the selection, which variables are in your model that you're clustering. So let's look at the data we've been analyzing in class and consider 2 of those. I'm going to look at sort of the variable selection aspect and the variable standardization and see how it affects our clustering of the data. So as a reminder, this data, we're looking at some knockdown experiments that were run, some gene expression arrays, So we're removing these proteins from our cells. It's a prostate cancer cell. After a couple days, we go in and we treat them with androgen, and we measure the expression right away, and then we measure the expression 16 hours later and see if we can find genes that respond to androgen even, and how they're different between these different treatment knockdowns. So here's the heat map of the gene expression data. So we have on the top, the dendrogram is clustering the samples, and on the left, it's clustering the features. And for this, I've included the 500 most variable features, and for no better reason than it's the number that we were using last week when we were talking about dimension reduction. So we can explore that choice in a moment, but right now that's what you're looking at. So the analysis steps to create this figure was that I standardized the genes first. Genes need to be standardized because on these microarrays, the values, the genes have different efficiencies, the reactions for measuring the different features, and they just are not on the same scale. So we want to standardize those variables. And then we're going to standardize the samples as well. I'll standardize both. And then I'm going to use pairwise, my similarity for both features and samples is going to be Pearson correlation. So the dissimilarity is 1 minus the Pearson correlation. And then my clustering criterion is Ward's distance, so that error sum of squares term. So this figure was reasonably robust. If I were to change the Ward's dissimilarity or the standardizing my samples, I'll get a similar result, I think. We can go check that in lab after this, in class this week. But I'll tell you one thing that did matter right now and show it to you, and it's the features. So I picked the 500 top varying, right? What if we have 47,000 on the array. What if we just took a random sample? So let's look at that. So here, the figure on the left is the exact same figure, just a little bit narrower, I think, than the one on the previous slide. But the one on the right, I'm just taking 500 samples at random, 500 features at random from the array. And well, you can see the difference immediately, right? There's no clear banding in our clustering. As far as the clustering at the top, it's changed. Although we do still see the largest signal, we do see a signal coming from the P300 knockdown group as having different expression than the rest, but the time effect, the genes, the samples are not separating by time based on the androgen. So we've lost those genes that are informative of the change of the hormone sensitivity. Those are not appearing in this map, and we're not seeing that because of the clustering. So the variable selection is important. What if instead we took the top 100 features and looked at how the effect of standardizing the sample variables or not? So that's what I did in this figure. And here I'm not going to compare the 100 to the 500 from the last slide, but let's just look at the effect of standardizing the columns between these two. So I have the exact same gene features, and I've standardized the genes the same way both times. Here I standardize the columns, and I'm using Pearson correlation. Here I did not standardize the columns, so I can use Euclidean distance for measuring distance on my samples. And now we see a difference. So it makes a difference whether or not you standardize your variables. Going to Euclidean distance, I actually get a very clear cluster here at the top, separating out the time effect between. So these are the 0 hour before androgen, before receiving hormone treatment. Here's after hormone treatment, and the P300 now, that knockdown set are not clustering together separately. But the larger effect is having had this time. And so it's the same genes in both analyses again, and the genes are standardized the same way in both analyses. But whether or not I standardized on the sample, the columns, before and here I'm using Euclidean distance, not the correlation measure, it actually brings out this cluster result. So know that there are these different ways of measuring distance, measuring the variables, and it will impact the groups and the clusters that you find. So what's right? Well, my remark is that if you have strong cluster signal, that's actually going to persist under many different algorithms and metrics. So whether or not you standardize or not, it'll persist. So you have to learn to recognize weak signals, I think, and be very careful of those, and then always try to validate and interpret your results so that you can tell why the result is meaningful and interesting.